So in this video, I'll be going into more detail with transcription and translation. So specifically in this video, I will only be going into detail with transcription, and the next video into detail with translation, just so it's not too much for one video. Bear in mind that I made a video explaining the Bob the Builder story, which is a very good analogy relating to transcription and translation. So before watching this, I highly recommend you go check out that story. That way, transcription, the more details will make a lot of sense. But I'll re-emphasize very briefly to those who already watched it as a reminder. So very briefly, we all know Bob the Builder, and we know that he likes to build things, right? So let's say one day he gets this idea to build a rocket. Now this is his house. In his house, he's got the study, he's got the garage, all those the common things, right? So what he needs to build this rocket is the knowledge, which he can get in the study, and the tools, which he always normally has with him, but sometimes he needs more tools, and the garage, the place to build it. So what he does first, what he needs to do first is first get the knowledge, right? So that he does in the study. So what he first does is he goes to get his book. He goes to the book, but he can't go into the book because this is his parents' study. They're working, so he's not allowed to go inside. So what he does is he asks them to pass the book, but unfortunately, they don't want to open the door. They're mean parents. So what they do is they're like, no, we can't give you the book. The door is locked. But he's smart. So what he says is, okay, how about you take the book and copy the page on how to build a rocket? Because this book is the book on how to build everything in the world. So only one page or one section of this book is going to teach him how to build a rocket. So he's smart, and he asked them to go copy that page. And now, since it's only a page, it can slide under the door. So the parents are like, okay, fine, you win. And so they go copy it, they get the page from here, and now it can slip under the door, and now he has it. Okay? So that's all that he had to do in the library. Next, so he needed the printer to do that, right? So he's got the paper now. Don't worry, just stay with me, because all of these things relate to obviously the cell and the real process. So just bear in mind, and this analogy will make it very easy to remember. So next, he goes, what he needs to do is, obviously having just a paper is not good enough. Because imagine Bob the Builder, he has a paper, that doesn't do anything. He needs many other things to build this rocket, right? Some things include his toolbox and some materials, right? So that's what he needs. So Bob the Builder is going to go get his toolbox and his materials, okay? So here he is. He's got the paper now that he needs, the instructions on how to build it. He's got his toolbox, and he's went to get his materials. So that's what he needs, and that will give him the final product, which is the rocket, right? This kind of all makes sense. Now, surprisingly, this matches exactly the process, the real process of transcription and translation. Let me show you. So translation, so this whole thing is the whole process, transcription and translation. This first part of turning the book and copying it to make a paper is transcription. The part turning the paper with some other materials and his tools into the rocket is translation. So this video will specifically go into more detail on specifically how the printer in the cell copies the DNA, which is the book, into mRNA, which is the thing you need, which is like the instructions to make the book, the specific instructions. Don't worry, I'll go into detail on that. So in the cell, how does this work? So the book represents DNA. DNA we know is found in the study of the cell, which is the library. All the information of the cell is in the nucleus, okay? So the nucleus is where we have the book. But obviously, just like the book can't fit under the door, the DNA can't fit through the membrane of the cell, okay? So what this body does is they also kind of copy a section of the DNA that they need, the only section that they really need to make what they want to make, or do the purpose that it needs to do. So the printer in the cell, or the photocopier in the cell, is Rna Polymerase. Okay, this is just the exact equivalent of the printer. It's going to go and copy that piece of DNA that it needs to be able to make the product that it wants. So now it has this right here. So the copied version, so the page that is made from the copying of the book, is called mRNA. Okay, you can see it's like half a DNA. It's like a section. Okay, this is the instruction manual you need. Now the last thing is to turn this mRNA, this instruction, into the final product. But just like in real life, we need tools and materials to make the final product from the instructions, so does the cell. And the things it needs is a ribosome and tRNA. Ribosome represents the toolkit, the tools that you're going to need to put it together. And tRNA represents the materials in the cell. Okay? If you don't understand it yet, just remember this for now, and I will go into detail in this video that will make everything make sense. This one and the next video. And having all of these components, so your paper, your ribosome, and your tRNA will give you the final product, which is normally going to be a protein or a polypeptide that will fix something in your body or make something important happen in your body. Just like the rocket, its purpose is to fly. So the purpose of this final protein will be whatever it needs to be in your body. Okay? So you can see how simple it really is. If you remember this analogy, remembering this is extremely, extremely simple as well. So let's go into transcription detail. So the only part we'll be focusing on in this video will be this part, transcription. So turning that DNA into mRNA, just as we saw here, DNA into mRNA. Now bear in mind, so this first part, like I said, happens in this room. This room in the cell represents the nucleus, okay? So far it's only in the nucleus. Whatever I'm going to talk about now is only in the nucleus. So first what happens is we know we have DNA. This is going to be now the more detail that you need to know. If you know all of these, there's nothing more you need to know, so just stick with me. So first we got DNA, okay? Just this thing. And what the body needs to do is it needs to now copy a little bit of it. How is it going to do that? So first it uses a little enzyme thing. This one you do not need to know. This is only for HL people. So just know there's a little enzyme thing, whatever. You don't need to bother about this, but I'm just going to explain it so it makes more sense. There's a little enzyme thing that's going to fold open a section of the DNA that it wants to copy. Okay? You don't need to know the name or anything. So it looks like this. So now what happened is this little enzyme thing folded open the protein so we can see a flat, because right now it's coiled. It's a helix. So we can't really copy. It's really hard. So what they do is they fold it open so you can see all the information. Okay? So now here we can see all the information, all the letters that make up DNA. And if you don't understand these letters, I really recommend you check out the video that I made on DNA structure, because otherwise this will be much more difficult to understand. So you need to know what this means already. So these are the letters. This is the letters that code for how you are, okay? Now, what are these little black lines in the middle? So these little black lines in the middle are basically hydrogen bonds. Hydrogen bonds, essentially what they do is they keep these little bases together. It's easy to remember that they're hydrogen bonds because the beginning is hide, and I remember it as like it's a hiding bond. It's not that strong. And that's true because right now you'll see these bonds are going to break so that we can copy apart, because it's really hard to copy information if they're all stuck together. So this hiding bond, this hydrogen bond, this weak bond, is now going to be broken in the next step. And bear in mind that this, just so you know, this orange part and this blue part here-ish, they represent the backbone of the DNA. So if you don't know the structure, here it is. But really go check out the video. So here you can see it's equivalent. So here's all the letters that we're seeing. Now this backbone, this orange part here, is basically just the sugar and the phosphates all linked together. And they're basically the thing that holds DNA together. So right here they're oversimplified. So they're not showing it, but just remember it is there. So this phosphate and the sugars are all lined up and linked together on both sides. But they're ignoring it and they're just showing the letters because that's what the important part is. Okay? So just so you know, just so it's not confusing. Yeah, so this part is there and it's represented in the orange and blue. Okay, so now the next part. So now that we've used this protein this enzyme to fold open the DNA, what we got to do is break these bonds. So what does that? So this one you do need to know. So remember how I said RNA polymerase is part of turning your DNA into mRNA? Now this is where it's useful. So this RNA polymerase is like a Pac-Man. It's going to go and eat the hydrogen bond. So it's going to go eat, and break it open basically. So now they will be separated. All these little bases that were bonded together by a hydrogen bond, which is weak, it's hiding, will now be separated. See, now they're separated. The DNA is now the same thing, it's just separated. And why is this important to separate it? This is super important to separate it because now it's possible to go in, for something to go in there and copy it. Because right here it was so difficult, everything was so cramped, right? So now the next part will be to actually go and copy the information. Now how does this happen? So luckily, you don't have to remember any new words, because the exact same thing that cut open, that broke these bonds, are gonna copy these letters. So how it does that is basically inside your nucleus, there's a whole bunch of letters floating around. These are the same letters here, but they're just floating around. So what this RNA is gonna do is it's gonna go and take these letters and go place them wherever they need to be to form a chain. And this chain will be this mRNA. Let me explain further. Remember how in DNA, A always binds with T and G always binds with C, no matter what. That's always how it works. So let me show you here. So in DNA, remember, adenine always binds with thymine, guanine always binds with cytosine. You will never see an exception to that in DNA. Okay? So now when we cut it open, now they're separated. And now we're going to use RNA polymerase. And if you watch my video on RNA, you will know RNA is slightly different. It doesn't use the same letters. It doesn't use A, G, T, and C. It uses A, G, U, and C. So there's no T in RNA. Instead, there will be a U. So in the cytoplasm, the letters that are floating around will be A, G, U, and C. There will be no T. Okay? So what that means is, and an easy way to remember that that which one binds with which is A and T are always together. A and T always together. Because if you know A and T are always together, then G and C will always be together because they're the remaining two. So basically now, using the same idea, A and T were always binding together and G and C were always binding together. Now this RNA polymerase will go and fetch this letter. It will go and grab it and bring it in there and bond it to where it's supposed to bind. So normally, T always binds with which one? T always binds with A, right? So what basically will, T always binds with A, right? So basically what will happen now is this thing will go and fetch an A and go and put it there. So here's an A, we can see it. So it's going to take this A, it's going to go and bring it there and place it right there. Okay? And then it's going to go to the next one. A normally binds with what? In DNA, A normally binds with T. But remember, in RNA that we're trying to make an mRNA, there is no T. So instead of T, this thing will go and fetch a U. So it's going to go take this U and bring it there and place it right there. Let me show you. So far we placed an A. Now we've placed a U. Now next what? C always binds with what? Yes, it always binds with G. So now it's going to go and fetch this G, bring it in there, and now it's a G. Now bear in mind that this process will now repeat for whatever specific section needs to be copied. So remember how I said for Bob the Builder, he only needs the page on how to build a rocket, right? So similar in DNA, when you go and copy or transcribe a specific part of your DNA, you're not going to copy the whole thing. You're only going to copy the part that you may need for whatever specific function your body requires. So in this case, this DNA, only this section will be copied, only this little section, because that's the part that's needed. So now this thing is going to bring all the necessary letters and bind them all along here until it's finished, until the specific gene has been copied. So let's say this is the part that needs to be copied, this whole part. So it's going to keep going and finish it. Okay? So now we have an RNA here, all these RNA nucleotides bound to the DNA here. Okay? So they're still separated, you can see here, but now we've got an RNA thing coming in here. So this is going to continue going until the specific gene has been transcribed. All the letters were added. All the codes were copied. Okay? So this is very easy to remember because in the printer, it copies the information. So that's how your DNA's information gets copied. Okay? This RNA polymerase will bring in the letters and basically this information will get copied into RNA. Now, one of the final things here, so it's going to go down now. It's got the final thing here. The final thing is filled up. And now, once the final thing is filled up, this little strand that has been formed, this RNA strand, will break loose. Okay? So it will basically, once it's formed, it will come out of the thing. So now it will be like this, see? AUG, AUC, AUG, AUC. So it was finished, and now basically it popped out of there and came out into the came out of the DNA. And now the DNA, these two strands here that I showed, these two strands that were separated, will now go back together like this. Okay, so it's basically, it was just used temporarily. It wasn't permanently changed at all. It was just copied, and the original DNA stayed the same after the process. After it was copied and the RNA came loose, the mRNA came loose, it was repaired and put back together. Just like the book. When Bob the Builder goes to use the book, he doesn't destroy the book. He photocopies it and the book stays the same. The DNA will stay the same, but now he has a page. Okay, now he has a page that will be needed to continue. So far, that was it. This is all that you need to know for transcription. How to turn that DNA into your mRNA. mRNA, the M stands for messenger RNA, and this is easy to remember because it is going to be the message that Bob the Builder is going to read and use his tools to build the final thing. Just like how your cell is going to use this message RNA to make the final protein or the final product. So I hope this all made sense. If anything didn't make sense, you can just write in the comments or anything like that, and I can try and fix it. But yeah, so overall, DNA into mRNA, that's transcription. It happens in the nucleus, and it's very simple, just using RNA polymerase to both cut the bonds, so cut open the bonds, and grab the little nucleotides of the mRNA nucleotides and build them onto the specific DNA section that you want to copy. Because by copying this specific section, you have now a message that just needs to be, that is going to code for what the specific DNA section coded for, the specific protein that it coded for. So next I will make a video on translation. So right now we made the message, now we need to turn this message along with ribosomes and tRNA, so the materials and the tools, into the final product. Just like I showed here. Turning the page along with the tools and the materials into the final product. Then we're done. Then we got the rocket. So make sure to go check this one out right after.